Thank you so much. I'm very excited, and I just realized that it's my first conference, international conference, that I'm speaking on aging. So I think a lot of the people in the crowd are not familiar with our work and what we do. So I was just thinking what I'm going to do in the next 15 minutes is just showcase some of the work in our lab and mainly the resource, the human resource and the biobank that we have. And hopefully I can get some, collaboration and people that can help us interpret some of the data. So we do have some people here in the crowd that are working with us and collaborating with us. And I think just referring to what Vadim said before and Andrew Teschendorf, for me, one of the challenges in aging is, or the main challenges, there are many, is the tissue specificity. So we know that we have, different tissues in human body, and I'll talk about it in a minute, and how does each tissue interact with each other, but also how does each tissue have their own specificity, methylation, proteomic, transcriptomic profile. Okay, all right, that was a big introduction. All right, so the overarching goals in my lab is to identify, or to try to identify, like everyone else, age-related epigenetic marks and pathways in multiple human tissues. We call it the Multi-Tissue Human Atlas Project, which I'll talk about it a bit in a minute. And then our strength is also exercise, so we're trying to identify markers that slow down the cellular molecular aging in males and females using exercise, and I'll talk about that. We've been really busy in the last few years accumulating and collecting from many, many data sets in our lab, around 90,000 omic profiles from epigenetic, transcriptomic, and a lot of proteomics recently in skeletal muscle across 18 tissues. That's a really big job for few people in my lab, across the lifespan from our lab and open access and other sources. All right, so central to that is the project that we're working on, which is exercise, and we just put it bioRxiv a few weeks ago, and the paper is now under review. On the molecular landscape of modality-specific exercise adaptation in human skeletal muscle through multi-omics. So it's a big title, but what we mainly doing is looking at the lifespan in terms of exercise and which specific modality changes which proteins, which DNA methylation, etc. We have a big study in our lab. It's called the GeneSmart, Skeletal Muscle Adaptive Response to Training. But we're also utilizing a lot of collaborators in Australia and in the world, for instance, the Welderly study, the TrainSmart, the testosterone, exercise and strength, EPH, and other studies that are providing us muscle samples, but also providing us data on muscle samples. Okay, and so it means that we have a biobank of around 3,000 muscle and blood samples with omics and exercise data across lifespan. That's a really big resource. And a lot of people are asking me, how do we do those exercise training and testing, and how do we take biopsies? So that's your time to shine now. Yeah, so what we usually do is putting someone on the bike, taking muscle biopsy, and then, next slide, please. And then we train them. It can be 4, it can be 12 weeks of high-intensity exercise. It's very controlled, so it's very hard to train those people because we need to control as much as we can with the nutrition, with the physical activity during those training. And we're also collecting a lot of physiological variables, or physiological measurements before and after training. So in a good year, we can collect around 20-25 people across this program, so it means that we need a lot of help from other labs, and we do get that because we have a lot of collaborators. All right, so I don't need to speak about DNA methylation. It will be embarrassing if I'll speak here on DNA methylation because everyone here in the crowd understands methylation much more than me, I guess. But we all know that there are two main ways, I guess, to measure methylation, which is EWAS and epigenetic clock, which has been discussed here a lot. All right, so what do we currently know about the aging methylome? quite a lot in the last 10 years. I think there's been, many studies talking about DMPs, variable methylation, epigenetic clocks, entropy, and correlation networks. And these are actually a few measures or a few statistical ways that we can measure DNA methylation. And most of the studies has been, and the clocks as well, has been focused on DMPs, differential methylation positions, which are positions that are changing average CpG sites, right? Okay, so this is the multi-tissue atlas of aging that we are trying to create in the last 6 to 7 years, I would say. It started with Christine Seal on the right-hand side. She was a talented PhD student in my lab. She finished her PhD last year. Then it continues with Max Way, who is a talented postdoc in my lab, and we are very much getting help from Andrew Teschendorf and other people in the crowd here. What we are trying to do is to find tissue-specific aging patterns, but also pan-tissue aging patterns across its already 18 tissues, across different methylation states. We also contributing to the BioLearn. This is a big project led by the Vadim Gladyshev lab and Jess Puganik and Madi Mokri. So we contributing samples to validate those age biomarkers. So I think it's a big and very important initiative. Okay, so DMPs, we talked about that. This is just chronological age, and Andrew, described it very, very nicely, how people are changing methylation and how this DNA methylation are really, really stable across the chronological age. VMPs are variable methylation positions, so it basically means that there is a site that can go up or down during aging. It's unpredictable. It's very variable, which is, we hypothesize that it's more reflective of biological aging. We don't know that though. Another measure is entropy, which is a single measure. So entropy is actually a measure of the amount of information in a set of CpGs. It's one measurement, which is really nice. And methylation becomes less predictable with age. We know that already from previous talk. And entropy is maximum if methylation is around 50%. So you can see here in the example, young and old person, and as we get older, we sort of tend to get more to around the 50% methylation. Correlation networks, which I'm gonna, talk about it, not talk about it today, but it's just co-methylation and the networks, and I think it's going to be presented in the next session. So this is highly correlated CpGs that are co-methylated. This is networks, and that enhance the biological signal. This is system biologies. All right, so the first chapter or the first paper from the Atlas we published this year with Andrew Teschendorf. This is just the blood, and you can just see the power of numbers here. So we use 56 whole blood data sets, 32,000 samples, and you can see here the change in average, the DMP. So 50% of the methylome is changing during aging, which is, it's a lot. It's really a lot. Then if we look at the variability, you can see how variable are a lot of sites. A lot of sites are very variable and unpredictable. And you can see that around 35% of the blood methylome is variable in methylation. But you can also see the really nice overlap between VMPs and DMPs. So sites that are both VMPs and DMPs. If you look at entropy, you can see on the green, you can see that those sites are sites that are DMPs that are driven by entropy versus non-DMPs in the gray. And you can see that there's across the meta-analysis, you can see that there is a lot of studies showing entropy is increasing in blood during aging. All right, so this is just some preliminary results, unpublished results that we are hoping to publish this year on few other tissues or multiple tissues. This is the brain. And what I want to show you in the next 3 slides, and this is new to me as well, so I'm hoping to get some help from you guys, is that how different the tissues are from each other in terms of the methylation changes during aging. All right, so this is the brain. We have 40 data sets, 7,400 samples, which is pretty impressive for brain in human. And what we can see here is, and I'll try to explain that without getting confused, is that you can see low methylation, high methylation, and intermediate methylation. Low methylation is lower than 25%, higher is higher than 75%. And in the inner circles, you can see these are young people, and they are being compared to old people. So what you can see here is it's pretty even. So people that have, or young people that have low methylation, you know, as we age, if we compare them to the age, if you start with low methylation, you can either go up or down in your methylation. The same with the high and intermediate methylation. So it's pretty even, the brain in terms of VMPs. In terms of DMPs, sorry. In terms of VMPs, you can see we identified 247 VMPs, roughly the same percentage of VMPs increase and decrease in variance with age. So for me, it suggests that maybe the brain is a tissue that the methylation is being balanced during aging. But again, I'm not really sure. In terms of entropy, you can see that there's a very small entropy, mostly driven by VMPs. You can see down here in purple, these are sites that are both VMPs and DMPs. Those sites are actually the most entropic during aging in the brain. Okay. Then, if you look at adipose tissue, and that's less samples, I guess, but still, you can see that, you know, young people, low methylation, 85%, but then most of this methylation is being increased with aging. So it's very different profile from the profile that I showed in aging. And in fact, you can see here that you have the high methylation. You have 10% of the people that have high methylation, and when they age, it's actually they get into almost maximum methylation, most of them, or 75% of them are increasing the methylation. In terms of entropy, there's a large entropy in adipose tissue compared to the brain, and you can see it here in both data sets that we show here. Okay, the last one is skeletal muscle, my favorite tissue in the body, and I'll talk about it in a minute in terms of exercise. And you can see here, very different profile. We have a lot of intermediate methylation, and 50% is positive, 50% is driven towards hypo and hypermethylation with aging. We don't have any VMPs that we found. Maybe it's the sample size, maybe there's biological explanation for that, which I don't really know yet. And then there's no entropy as well. Okay, so let's talk about skeletal muscle in the time that I have, which is my favorite organ in the body. We're working on skeletal muscle for many years. And this is just an editorial that we recently written about exercise and aging and biomarkers, right? So skeletal muscle is 40% of our body, so it's very important, and it initiates the movement and life and oxygen. At the physiological level, aging directly impacts skeletal muscle. We know that, but the good news is that we can train the muscle even if we are 70, 75, and we haven't moved all our life. So it's very elastic, of course. And so there's never a late time to start an exercise. There's a global effect of exercise on various molecular pathways. All right, so a few years ago, we built an epigenetic clock, a first-generation clock with Steve Horvath on skeletal muscle, the first one in humans. And as we add more and more samples in humans, as we get in more and more samples, we can see that the chronological age is really, really tightly associated with the predicted age, which is really nice, but it doesn't insinuate any biology for us at the moment. All right, what I want to show you lastly is data about exercise, data that we published last year in Aging Cell. This is driven by Sarah Voisin, who was a very talented postdoc in my lab. Unfortunately, she left to work at the Novo Nordisk in Copenhagen, which I can totally understand. So, high aerobic fitness and exercise training are associated with younger methylation profile. As you can see here, this is just a correlation plot. You can see here we have the age, we have the fitness, and when I say fitness, what we measure is VO2 max, maximal oxygen consumption at baseline. We measure it a few times to be sure that what we do is we capture the real VO2 max and not noise. And exercise response, so what we did is we looked at people after exercise, so the response to exercise. So you can see here the age is correlated or has inverse correlation with both fitness or VO2 max, which is a measure of aerobic capacity, but also with the response to exercise. So those CPG sites that are changed with age or hypomethylated with age, what happens with exercise? Even exercise of 4 weeks of high-intensity training, it, I would say, revert or taking back the methylation to almost the same stage that they were before. Not quite the same, but it takes it back to the same methylation as they were before they train. In terms of that, so we've got, and we also have like a nice positive correlation between the fitness and the exercise training. Okay, so from those 3,000 CPG sites, age-related CPG sites that we narrowed down, you can see it here just in heat map, the VO2 max, the age, and the training, and you can see the sort of the nice trend, inverse trend between them, especially between age, VO2 max, and training. All right, transcriptomic, we did the same. We were really lucky because we also had one cohort of DCUs, which means people that for some reason they put a cast on them or put them to bed for a few weeks and they didn't move or they didn't do any physical activity at all, right? And their profile, their CPG profile looked exactly like someone who is aged. Whereas there was the same correlation with age in fitness, age in exercise training, was also negative correlation as we expected. So that's just giving us some hope that exercise is very helpful in methylation and proteomic, in transcriptomic, and we're looking at proteomic at the moment. That's just the same heat map. All right, so with that, I would like to thank you so much for listening to my talk today and acknowledge all the other people that are hard workers in my lab. Thank you. Yeah.
Thank you very much. Now open for questions. Yes.
Thank you very much for such great work, amazing data sets. I was wondering regarding the part that is yet unpublished. Will you still publish the data itself? Will it be open?
Yeah, definitely. So one of the things that we reiterate in our lab is that we put everything in bioRxiv. We put all the data in open access in GO, and everything will be open to the public. That's our contribution to the science, and I feel like that's very important. Yes.
Brilliant. Thank you very much.
Next question.
Thank you for the great talk. Sorry. Have you measured the epigenetic age before exercise and after exercise of the same individual?
Say it again. Of the same individual?
The same individual. Did you measure epigenetic age before exercise, the exercise program, and after the exercise program?
In terms of clocks?
Between 4 weeks, or that you...
In terms of, like, in clocks?
Yes,
epigenetic age in clocks? We've done that. We really haven't found anything that we can report.
Oh, thank you.
Thank you for your excellent
Thank you
talk.
so much for your very enlightening talk. Could you flash back to your slide on obesity?
Obesity?
If possible. So my question is whether you could enlighten us what we have found so far about obesity, because according to the ACSM protocol, exercise is medicine, we need about 300 hours of exercise per week, and they found it's quite difficult. And according to the latest medical journal, the consensus seems to be like we need to combine exercise with GLP-1 agonists. And that's why even in Singapore, Ozempic is like hundreds of dollars beyond the reach of most people and not subsidized. So perhaps you could enlighten us that would address that financial problem of people with who are obese.
So I couldn't agree more. So we're still trying to understand the adipose tissue and what does it mean in terms of DNA methylation. But I couldn't agree more with what you said.
Okay,
thanks.
Question here.
Thank you for your talk. Remember in your talk you mentioned you do the multi-tissue atlas, including the brain, adipose tissue, and skeletal muscle, right?
Yeah, that was
have you ever
just
considered
examples
about the linkage between these tissues during aging? Because we all know like muscle during aging, they got sarcopenia, they got the adipose tissue invading, they got the neuron degeneration. So have you tried in your system to consider about the talk between these tissues?
Our next stage, and that's exactly what I said before, our next stage, and that's what we're doing now, we're trying to find common sites between multiple tissues. For instance, one of the work that we are just doing is trying to, and that's a big work, that's a big project of Maxwell Jacks for my lab. We have now connections with a few hospitals, and we're taking cardiac muscle from people who are undergoing surgeries or from deceased people, and we want to see the common between cardiac and skeletal muscle, for instance.
Thanks.
Yes.
Hi. When you look at certain physiological traits like VO2 max, exercise can improve it.
Yeah.
You still decline.
Yeah.
And then there's other physiological traits like maximum heart rate.
Yeah.
Exercise can't improve that.
Yeah.
So it's a kind of slowing down, you can say decline, but in a chimeric way or a partial way. So I wanted to ask you, what parts of the decline exercise do you think can slow down, and what other parts it can't?
In terms of what? In terms of the tissues?
Physiology.
Physiology. So I think in terms of function, I think functionality is one thing that I think it can slow down. So just, when you're 70 or 60 or 75 or whatever, if you can just, walk, if you can be mobile, if you can, sit and properly and get up from the chair. So I think the functionality is really, really important. I think the oxygen consumption, time is up, the oxygen consumption is really, really important as well in terms of physiology. So the more oxygen you have, the better when you're old. But I guess the only thing is that, there are traits that can be changed and there are traits that are signature, like maximal heart rate. So hopefully I answered the questions. Thank you so much.
Thank you very much. Thank you, Professor Eynon.